Bismillahirrahmanirrahim. Different DNA motifs that bind these proteins, they can not only bind to the DNA but also the other proteins of so as to activate the transcription. In the DNA binding domain, there are different three types of motifs usually seen. One is the zinc finger, helix turn helix, and leucine zipper. The eukaryotes, they show the zinc finger as well as the leucine zipper DNA motifs or DNA binding domains, while the helix turn helix, this domain is shown in the prokaryotes. Three different types of DNA binding domains present both in prokaryotes as well as in eukaryotes. Prokaryotes show helix turn helix mostly, while the zinc finger and leucine zipper DNA motifs, they are present in eukaryotes. By means of these motifs, the protein they can bind with the DNA. This is the zinc finger motif. A zinc finger motif contains alpha helical structure as well as a loop structure. Zinc is present here. The zinc molecule, it is stabilized by two cysteine residues and two histidine residues. This one is the alpha helical structure of the zinc finger and loop structure. All of these combine, and by means of repetition it can bind on the DNA. This is the DNA, and the protein, they can bind on the DNA by means of this zinc finger. This is the helical structure, repetition of the zinc finger, and the loop structure. The helical structure fit into the major groove of the DNA. By means of, in eukaryotes, this zinc finger, it is most common in DNA binding domain. Leucine zipper, its appearance just like a zip, so it is known as the leucine zipper. Here the helical structure has been seen, and it can fit themselves into a DNA. Leucine zipper is also present in eukaryotes. This is the helix turn helix. Helix, then this one is the turn. The amino acid sequence, they are arranged themselves in a turn and then helix. So it is known as the helix turn helix. This helix turn helix is most common in prokaryotes. The lac operon, the repressor, it can bind with the operator region through this helix turn helix structure. Helix turn helix structure, it is mostly common in prokaryotes. Now the gene regulation in eukaryotes. In prokaryotes, genes, they are mostly regulated at the transcriptional level. Whether they halt or restrict or inhibit the transcription, or premature transcription take place, premature attenuator take place, or whether they, through the repressor region, it can turn on or turn off the operon. But operons are absent in eukaryotes, and instead of operons, the eukaryotic messenger RNA contains monocistronic. Only one gene can be transcribed into a coding region of eukaryotic messenger RNA. Along with this gene, it contains the regulatory sequence in eukaryotes. The regulatory sequence consists of enhancers, the promoter region. Promoter region may be a proximal promoter region or maybe a distal promoter region. And these promoter region, and they can bind the general transcription factor as well as the specific transcriptional factors. The response transcriptional factors bind to the, or the activators bind to the enhancers. Enhancers are the nucleotide sequence. They are thousands of base pair away from the transcriptional start site. So these are the nucleotide sequence that they are also known as the response elements that can initiate to activate any gene. And also they contain the promoter region, proximal promoter region on which the general transcription factors they bind, and the distal promoter region on which the specific transcription factors they are bind. For the activation of transcription, there must be interaction between the transcriptional factors bind on the proximal promoter region as well as the distal promoter region, and along with the activators through mediators. So this interaction it has been done by the bending of the DNA. DNA bending proteins, they are responsible for bending, and also they are responsible for carrying this interaction between the activators and the transcriptional factors through a mediators. So different promoter region as well as the enhancers, all of these, they are the nucleotide regulatory nucleotide sequence. They are used to regulate the eukaryotic gene expression. There are different level of gene regulation of gene expression. It may be regulated at the transcriptional level, but the primary transcript it undergoes the post-transcriptional changes, so it may be regulated at the post-transcriptional changes. In the cytosol, there is a translation. In the eukaryotes, the post after the post-transcriptional modification, then the translation it is started. So in the cytosol, when translation has been started, then it undergoes the further post-translational changes. So all of these, they are responsible to mediate the regulation of gene expression, all of these levels. So in eukaryotes, this regulation it is much more complicated as compared to prokaryotes. One of the example is the coordination regulation, in which, since the eukaryotes they contain more than one chromosomes, and these chromosomes they contain the genes for the same response. So how all of these genes they can be regulated at the same time, this has been done through coordination regulation. The specific transcription factor present, they regulate the same gene. For example, in the galactose in yeast it is the unicellular, but it is eukaryotes. This yeast, in the absence of glucose and in the presence of galactose, it always binds the transcriptional factor at gal4. This is the gal4 transcriptional factor. It binds on the upstream activating sequence. Upstream activating sequence is present at the upstream of each gal gene. The gal gene, they are responsible, they produce the products which are responsible to metabolize the galactose. Gal4 always it bind to the UAS upstream activating sequence whether galactose is present or whether galactose is absent. In the presence of galactose, what happened? The gal3 um, protein it is activated, and this gal3 protein it bind with the gal80. Gal80 bind when there is a presence, there is absence of galactose. Gal3 is activated when there is a presence of galactose. This gal3 it bind the gal80 and detach this complex from this gal4. Now gal4 is responsible to activate the transcription by incorporation or by recruitment of this saga, which is the um, uh, this one is the another transcription factors and data binding proteins. So for the recruitment of other transcriptional factors, this gal4 will be activated when there is a detachment of gal80, and this gal80 has been detached from this gal4 when it complex with gal3, and gal3 is activated only in the presence of galactose. So this one is one of the example of coordination regulation. The GAL4 protein, it binds to the DNA through a DNA binding domain. Two domains present this GAL4. One is the DNA binding domain through the zinc motif. It can bind on the DNA. But the other side also contains the termination activation domain. This transcription activation domain, not termination, transcriptional activation domain, it can bind the other proteins just like a gal-80. Similarly, gal-3 and gal-80, they contain the transcriptional activation domain by which they can interact to each other, and also gal-80 contain the transcription activation domain by which it can also interact with the gal-4. So these different domains, they can bind not only to the DNA but also bind with each other. Another example is the hormone response system. Hormones may be a peptide in nature, or maybe they are the steroidal in nature. Steroids hormones then can easily pass through the plasma membrane, enter into the cytosol or nucleus, and it can bind with their specific receptors and form the complex. This one is the steroid receptor complex. This is the general mechanism. Now these receptors, they are basically the transcriptional factors, but they can only be activated when they bind with their respective ligands. These receptors now contain the three types of domains. One is the DNA binding domain, other one is the ligand binding domain, and third one is the transcriptional activation domain. When they bind with their respective ligands, now they being active, and now they bind to their response elements. Response elements, they are the nucleotide sequence present on the DNA. For example, glucocorticoids. These are the GRE, glucocorticoid response elements, or enhancers. These glucocorticoid response elements, when bound with these activated transcriptional factors, it can now lead to the or mediate the response for the activation of transcription, and so the production of the protein. This one is the example of the steroid hormones that contain the intracellular receptors. But the other type of receptors, they may be peptide in nature, can't cross the cell membrane. How they mediate their receptors? When they bind with their respective receptors, for example, the insulin hormones or glucagon hormones or epinephrine, norepinephrine, all of they are the peptide in nature. When they bind with their receptors, what happen, their intracellular, they mediate the reactions by which the cyclic AMP, it be, for example, the example of the glucagon, then the enhance the concentration of cyclic AMP, it will be enhanced, and this cyclic AMP, then this it will bind to their it will also phosphorylate or activate its protein, which is known as the cyclic AMP response element binding protein. When this protein CREB protein, when it will be activated, then it will undergo to the, it go towards the nucleus, from where it can bind with this response element, the CRE, cyclic AMP response element binding response element. CRE is basically the nucleotide sequence present on the DNA, and when it binds with the CREBP, the cyclic AMP response element binding protein, it can now activate the transcription. So the peptides, hormones, or the which can't cross the cell membrane, they can also mediate their response through the transcription. The CRE is the response element dependent on the cyclic AMP concentration, and the CREBP, they are the response element binding protein. This one is the response element binding protein, which activate when there is a high concentration of cyclic AMP, and cyclic AMP is formed when glucagon, it bind with its receptors.